Welcome back to the deep dive. So, picture this challenge. You want to map the human brain,
Gotcha.
but you need a map that's detailed enough to see layers of cells, yet, still shows the entire three-dimensional organ.
It's a classic problem. The zoom in versus the big picture.
Exactly. We've had brain maps for years, but they're either super high-resolution images with no labels, or they're like these low-res roadmaps of the whole country. So the big question is, how do you get that microscopic detail, the cytoarchitecture, while keeping the whole 3D atlas together?
And that is the fundamental hurdle neuroimaging is trying to clear right now. I mean, when researchers compare brain scans, say, for Alzheimer's or even just healthy aging, they need a reliable standard grid.
A common coordinate frame, or CC?
Precisely, a CCF. And within it, they need to segment regions of interest, or ROIs. But the existing tools, well, they just gave us these coarse, fuzzy boundaries. But now there's a new development that really promises to bridge that gap. It's called NextBrain.
NextBrain. Okay, so our deep dive today is going to be all about unpacking this probabilistic histological atlas. We're going to get into the weeds on the custom AI they used to build it, and then show you why its granularity is, well, it's really starting to revolutionize how we analyze disease and aging.
And to really get why this is such a big deal, we need to clarify a couple of terms right up front. First, histology.
Okay.
This is the absolute key. It's not an MRI. Histology is where you take the actual brain tissue, slice it incredibly thin, stain it with dyes, and then look at it under a microscope.
So you're looking at the real cells.
The real cells. That's what gives you the cytoarchitecture, the cellular detail, that even a super powerful ex vivo MRI, which might top out at 100 micrometers, just can't see.
But wait, histology gives you the detail, but it literally breaks the brain up into thousands of little flat pieces.
Exactly, which brings us to the second key term, probabilistic labels. Because NextBrain is built from several different brains, you don't just get a single hard line for a border. Instead, for every single tiny point in that 3D space, the atlas gives you a probability, a likelihood, that this point belongs to, say, the hippocampus or the thalamus. And this probabilistic way of doing things is, well, it's absolutely vital for training accurate automated tools.
Let's unpack that a bit. Why is NextBrain so necessary? I mean, we have big established tools, FreeSurfer, FSL, SPM, that are used in huge studies. Why couldn't they just deliver this kind of detail?
It really boils down to the source material they were built on. Most of those atlases are derived from averaging in vivo MRI scans, living people.
Right.
And they operate at about a 1 millimeter resolution. And a millimeter, when you're trying to map tiny, distinct little subregions inside the thalamus, it's huge. It's way too coarse.
So previous attempts to go high-res, they must have fallen short somewhere. What are the big issues?
Yeah, there were a few major efforts, and NextBrain really had to overcome their limitations. First, you had Big Brain.
I've heard of that one.
Incredible detail. It's this ultra-high-resolution reconstruction of a single brain. But, and this is a huge, it was published without anatomical labels.
Yeah.
It was like having a beautiful, detailed map with no street names, no cities, nothing.
A map without a key, useless for navigation.
Exactly. Then you had Histological brain. It pulled data from 23 different people, mapped 248 areas. Better, but researchers found it just had limited accuracy, especially in those deep subcortical areas. And finally, the Allen reference brain.
Also very famous.
Very. It has comprehensive annotations, but they were only applied to a sparse set of sections from just one specimen. It wasn't a dense, fully 3D map you could use for segmenting any one scan.
So we had detail but no labels, volume but not enough accuracy, or labels that were just spotty.
Right, and that's the gap NextBrain fills. It's designed to deliver all three.
Yeah.
Densely labeled 3D histology of the whole brain and the probabilistic atlases you need.
Which is how we jump from Allen's 138 regions to NextBrain defining 333 regions of interest.
A huge leap. 34 cortical and an incredible 299 subcortical.
The ambition there is just wow. And the process to build it must have been equally ambitious. The analogy I loved from the sources was imagining a 2,000-piece jigsaw puzzle, but in 3D.
Yeah, it's a good one.
And the pieces are paper thin, they're warped, they're torn, they're cut at weird angles. That's the reality of reassembling 10,000 histological sections from 5 brains. So how on earth did they use AI to solve this puzzle?
They had to build a completely custom 3-part AI framework.
Wow.
Off-the-shelf tools would just fail. The first part is something called the differentiable regularizer.
Okay, what does that do?
So when you slice the tissue into blocks for processing, you get these tiny gaps or sometimes overlaps when you try to digitally put them back together. The regularizer is like a mathematical glue. It minimizes those gaps and overlaps, making sure all the block boundaries meet smoothly in 3D. It makes the final map continuous.
So that handles the physical errors from the slicing itself, but then you have the alignment problem. You've got this beautiful, detailed histology image, and you have to match it to an MRI image, which looks completely different.
That's the hardest part, and it's solved by the second component, contrastive artificial intelligence registration, or synth by red.
Synth by red.
Think of it as a style translator. Histology has all this rich texture from the stains. MRI is all about gradients and contrast. Synth by Reg uses a deep learning network to perform, well, style transfer. It synthetically makes the histology image look like an MRI slice.
Wait, hold on. So the AI puts on a digital disguise. It doesn't change the anatomy. It just changes the texture so the computer can match them up.
That's it exactly. It's a wildly clever solution. Once they share a similar visual style, the system can align them with incredible accuracy.
Okay, but what about distortion? If you're stacking 10,000 slices, one tiny error in alignment must get magnified over time.
It does. And that's what the third and most mathematically intense component prevents. A Bayesian refinement technique based on Lie algebra.
Lie algebra. Okay.
You just align slices sequentially, 1 to 2, 2 to 3, and so on. You get this infamous banana effect.
The banana effect.
Yeah. Structures that are naturally curved, like the hippocampus, they get unnaturally straightened out in the final 3D model.
Because each slice is only looking at its immediate neighbor.
Exactly.
Yep.
This technique, using Lie algebra, ensures 3D smoothness across the entire volume at once. It forces every section to align not just with its neighbor, but with the whole 3D MRI reference, preserving that natural curvature.
The technical sophistication there is just staggering, and the numbers back it up. The sources show this new method cut the average registration error down to just 0.99 millimeters.
Which is a huge improvement from the previous best of 1.44 millimeters.
That's the difference between a blurry boundary and a crisp, definable structure. So we've seen how they built this. And for you, the listener, knowing the incredible effort that went into this, what part of the brain are you most excited to see mapped with this kind of detail?
So now let's shift from how it was made to the final product itself, the probabilistic atlas. A key point is that NextBrain was built from only 5 hemispheres.
Right. 3 right, 2 left, and the right ones were flipped to make pseudo-lefts. That sounds like a really low number. It brings up concerns about bias, right?
It's a critical point, yeah. If the map is based on just 5 people, how do you know it's not just showing the quirks of those 5 brains instead of general human anatomy?
So how do they solve that?
They solved it by anchoring the whole thing to an established standard. The labels from the 5 hemispheres were merged, and then the entire atlas was registered to the MNI template.
The Montreal Neurological Institute Atlas.
Exactly. And the MNI is derived from 305 subjects, so it's a really robust standard frame. By warping NextBrain's data into that MNI space, they basically center the atlas and prevent that individual bias.
And the final map is it's an incredible leap in granularity. We are talking 333 ROIs on a 0.2 millimeter isotropic grid.
The difference is night and day. I mean,
Right.
think about the Samsung Atlas and FreeSurfer. It gives you 13 ROIs at 1 millimeter resolution.
13 versus 333.
It's a different world. With NextBrain, you're getting dozens of distinct subregions for the thalamus, the hippocampus, the amygdala, structures that are just so central to things like memory and emotion.
And the best part is, it's actually accessible now. The Atlas ships with a Bayesian segmentation tool that's integrated right into FreeSurfer.
Which means any researcher can take a standard MRI and in about an hour on a decent computer with a GPU, segment all 333 of those regions automatically.
It just democratizes this incredibly detailed analysis. You don't need a team of highly trained neuroanatomists anymore.
And that brings us to the so what part. The applications that really show the power of this detail.
Let's start with the big one, those massive ultra-high-res scans.
Right. Think about those 100 micrometer whole brain scans from a 7 Tesla MRI. They can take 100 hours just to acquire. You end up with a data set of over 1,000 slices.
Manually segmenting that would be
Impossible. It's practically infeasible. But NextBrain automates it, and the validation showed it works really well with a median dice score of 0.667, even for tiny, tiny nuclei. It proves you can automatically localize even the smallest deep brain structures.
the clinical impact, though. That's where it gets really interesting. How did NextBrain do when it was used to classify Alzheimer's disease?
This is where the granularity really paid off. In a test using ROI volumes to classify patients, NextBrain achieved an area under the ROC curve and ERO-OC of 0.953.
So about 90% accuracy.
90.3% accuracy, yeah. And that significantly outperformed the Allen M&I template, which got 0.929. The fine-grained detail of the 333 ROIs just provides a much richer, more distinct signature for the disease.
That's powerful. But for me, the most revealing thing was what it uncovered about aging. Using the Aging Human Connectome Project dataset, it showed that aging isn't just this global, uniform volume loss.
Not at all. It's targeted. NextBrain found these differential aging patterns within single structures.
Yeah, like the caudate.
Exactly. Look at the caudate. The anterior part showed a much stronger negative correlation with age, so more volume loss, than the posterior part. Same thing in the globus pallidus. The external segment showed a stronger age correlation than the internal one.
So we need to stop just saying the caudate shrinks and start asking which part is shrinking and why. Is it tied to specific brain networks?
That's the strong implication. And in the thalamus, the areas that declined most were the mediodorsal, anteroventral, and pulvinar subnuclei.
Which aren't just random spots.
Not at all. They are key nodes in our limbic and prefrontal circuits, the ones critical for executive function and emotional regulation. It confirms that brain aging is highly network dependent.
The ability to see that different parts of one structure are aging at different rates, that really shows how NextBrain lets us map specific network decline. It's a public, customizable resource that just opens up a new world for researchers.
And it lets the whole community really leverage the data coming from newer technologies like 7T MRI. It just increases the specificity of what we can find in both health and disease.
So the atlas shows these very specific anatomical differences related to age. It shows atrophy isn't a blunt instrument, it's a targeted process.
Which leads to our final question for you to think about. Considering these new localized aging signatures, what existing assumptions we have about global brain atrophy should researchers now immediately start to revisit? That's the foundational question this new atlas can finally help us answer. Thank you for diving deep with us.